Okay, third time's the charm. My name is John Williams, and my presentation is entitled Gut Microbiome and Epilepsy. There's an issue with sound with the last recording. Hopefully, that's remedied now. Okay, epilepsy affects 50 million people worldwide, with 125,000 passing away of the illness every year. Epilepsy is a neurological disorder characterized by repeated seizures caused by sudden and temporary changes in the electrical signals in the brain. These can come about through stress, hunger, lack of sleep, lights, various other reasons. Treatment is usually drug-resistant epileptic drugs. Sorry, that takes care of most cases. In extreme cases, surgery is necessary. Vagus nerve stimulation, or even a ketogenic diet. Ketogenic diet is usually done with children, and unlike current ketogenic diet, in which it is practiced for weight loss and a host of other reasons, ketogenic diet done to prevent seizures has a much higher fat content. Gut microbiome is located in the large and small intestines of the human digestive tract. Well, actually, all other animals also have various makeup of bacteria, fungus, viruses that are in the human digestive tract. Some of them if they have an overabundance, can cause illness, and they're usually in a symbiotic relationship with the organisms where they're housed, sometimes helping to provide nutrients such as vitamin K or short chain fatty acids by digesting materials that we cannot digest. Diet, medication, and environment influences the makeup of one's gut microbiome. Also, we get an initial host of gut microbiome through vaginal birth and breastfeeding. And there's been study ongoing that is fairly recent, in which they're studying how gut microbiome affects human health, mentally, physically. And in this case, we're talking about, I guess, brain health and how the influence it has on epilepsy. Purpose of this meta-analysis is to examine recent research that highlight differences in gut microbiome between drug-sensitive, drug-resistant epileptic patients and healthy controls, in hopes of finding a common set of bacteria that may lead to possible avenues of treatment. This is an attempt to find a consistent pattern of bacteria that is present in the human gut that may hopefully point to clear indications of what is consistent in epileptic patients versus healthy controls. The methods I use: Google Scholar, and of course Auraria's Campus Library search engines to find research using such terms as epilepsy gut microbiome research, epilepsy and gut microbiome, the effect of gut microbiome on epilepsy, and epilepsy gut microbiome. Exclusion criteria: studies that involved animals are just focused on humans. They can induce epilepsy, of course, in animals, and study the effect of changing diet or what bacteria is consistent in their gut. But I just wanted to restrict my search in humans. 2, studies, of course, before 2018 were also excluded. Studies that were meta-analysis or reviews, and of course case studies. Inclusion criteria were, of course, studies that were 2018 or later. I began this in 2023. I know it's 2024, but I just kept the 2018 number, especially with so few studies found. fecal samples that were analyzed using the 16S rRNA or the 16S DNA sequencing data. These were just used to determine what the bacteria that were found was using these sequencing techniques. Included also were epileptic patients who were compared to healthy controls, and epileptic patients who were drug sensitive compared to drug resistant patients, and research that involved child or adults. This was due to the sample size of the available research being so small. And here's how things broke down. Epilepsy and gut microbiome research. When I say studies, that included animals and humans. Animals especially seem to be more abundant. So I had to cut out animals. That got me to about 25 studies just involving humans. And then I had to cut out reviews, meta-analysis, case studies, and leave myself with random control trials. That got me that got me to 9 studies just involving humans that are random control trials. Case studies were interesting, but they were anecdotal studies. For instance, a patient's Crohn's disease who had Crohn's disease and epilepsy and changed their gut microbiome through fecal transplant to treat their Crohn's disease and found a remission of their epilepsy. Interesting, but it was only one case. Methods. So as I said, I got 9 articles, 5 done in China, 2 done in South Korea, 1 done in Turkey, and 1 from Italy. As I said, I took all those studies and I looked at the gut microbiome that was found on a genus level that were common to all of them. Unfortunately, there were no bacteria that was found in epileptic patients that were common in all the studies, unfortunately. So the results. Studies that allowed the best opportunity, as I mentioned, for comparison were those on the genus level. All studies except one reported back bacteria that were found on the genus level. And the one study that did not, it has species level. So I just went from there and trying to connect that to the other studies in terms of connecting them to their genus, those species. Okay, 2 studies out of China and 1 out of Turkey found an abundance of Fusobacterium in epileptic patients. And 1 China study has a greater abundance of bacteria found in drug-resistant patients. 3 Chinese studies and 1 Korean study showed an abundance of Bifidobacterium in patients with epilepsy. However, there was one Turkish study that showed a statistically significant abundance of that bacteria in healthy controls rather than epileptic patients. So that was interesting. And one Korean study, in that Korean study, that was only found in 3 of their 8 intractable epileptic patients. 2 China studies revealed an abundance of the genus Escherichia shigella, pardon me if I mispronounced that, in the gut microbiome of epileptic patients. Excuse me. 1 found the enriched abundance of that bacteria in pre-treatment, in the pre-treatment group, and a reduction in the post-treatment. However, there was no significant difference between post-treatment and healthy controls. A separate study had an abundance of this genus in healthy controls over those found in epileptic patients. Again, a contradictory indication. 3 China studies found an abundance of Prevotella and Megamonas in epileptic patients or healthy controls. First study found these genus part of a group of bacteria that comprise 50% of gut microbiome in healthy controls and epileptic patients. However, there was a greater abundance of this in the healthy controls. Second study found an increase of Prevotella on the species level. And in this study, Megamonas was less abundant in epileptic patients in rural areas than patients in urban areas. That's the one study that did urban areas versus rural areas versus healthy controls. And third study found the abundance of Prevotella megamonas in epileptic patients. Another study found Prevotella 9 was almost non-existent in drug-naive epileptic patients, but increased after admission of anti-epileptic drugs at the 4-month point and the 12-month point to levels similar to that found in healthy controls. And a Turkish study from Bacteroides was significantly higher in the healthy group than in the epileptic group. The Chinese study found significant difference in abundance of this bacteria between epileptics before and after treatment in the control group, or no significant difference, sorry. And another Chinese study found that Bacteroides fragilis was positively correlated with epilepsy. Again, contradictory statements about the species and genus level, how they are affected. And a Korean study found an abundance of Bacteroides finding Goldie was increased in drug-responsive epileptic patients. Excuse me again for my pronunciations. Conclusions and limitations. As I mentioned, there is no clear specific bacteria profile or certain bacteria that would be indicative of epileptic patients. Again, this study may be too small. These studies may be too small. They range from 14 patients to 156 patients with a mean of 62.2 patients. Some of them are more, of course, more statistically significant than others, but perhaps a much larger sample size was needed. 50 million people with epilepsy worldwide. Maybe they should have taken longer, or it's suggested that they take longer possibly to get a larger sample size, see if that has an impact on the bacteria they find. But it's, of course, hard to say without the actual research. And of course, it seems like some of the biggest differences were because of geographical location. Like some of the similar studies were in China. Of course, there were some that were dissimilar that was also in China. When we compare China with Turkey, that was a pretty decent difference of bacteria profile that was indicative of epilepsy found between those two countries, China and Turkey. So that could be an issue also. And also, there could be various combinations of bacteria that may be attributed to or associated with epilepsy that are too complex for these studies to determine. So those are a bunch of possibilities that could be the result. But it's so complex and we're so far at the beginning, we're so into the beginning of this that it may take a while and many more studies to figure this out. Okay, and that's the end of it. Came up just short of 15 minutes. Hopefully sound recorded better in this one. So we shall see. Thank you for your attention.